Okay, hi everybody. We are starting again with scientific methods. And so this is going to be the second section on your page, and it's actually got a typo, and it says 3 scientific methods. So that's where we're starting. So we talked about what science is, and basically that it's the way that we study the natural world, and the scientific method is the way that we do that. So we're going to talk about that now. So the scientific method itself is a series of steps that scientists use to solve a problem or to answer a question. So we talked last time about how science is solving a problem and answering a question, but the scientific method is the steps to do so. So the steps go like this. We have an observation of something. We see that something's happening. We ask a question about it. We form a hypothesis, so our idea of why this might be happening, and then we design an experiment to figure out if that is why this is happening. Once we do the experiment, we record and analyze the results from it, and then we draw conclusions based on that. So let's go more into detail here, and you're following along in your notes, hopefully, writing these things down. Remember, pause anywhere if you need to. So step 1 is making an observation or asking a question. So we have to figure out what we want to figure out first. So an example of that might be how much water can a root hair absorb? So those little tiny pieces that come off of roots, if you've ever gardened or anything like that, that's what they're talking about. Why does a plant stem bend toward the light? Might be something that we identify. Or what effect does temperature have on heart rate? So these are all things that we might ask or we might notice, and we want to figure out why these things happen. That's the very first step. Step 2 is to form a hypothesis. So a hypothesis is a possible explanation to one of those questions. So first we ask a question, we observe something's happening, and then we figure out what we think might be why. So it's just a prediction. We haven't proven anything or disproven anything yet. It has to be stated in a way that we can test. So a hypothesis can't just be a question. We have to have a way that we can test it. And it's considered testable if evidence can be collected that either does support it or does not support it. So we have to be able to decide one way or the other. Remember, go back and pause if you need to. So step 3, once we figure out what we want to look at, we predict why something is happening the way that it is. Then we have to design a controlled experiment to either support our hypothesis or disprove it. So there's a lot to an experiment. So these are very important things. So the factors in an experiment that we can change are called variables. And we'll use that word a lot as we go through our class this year. So some examples of variables would be changing the temperature of something, the amount of light that we have, like if we think back to our question in the previous slide of why does a plant stem bend towards the light? We might figure something out based on what we know from last year about photosynthesis, and that it needs the sunlight for something. So we could design an experiment that would test the amount of light that different plants would get, and that would be a variable that we're changing. Time's a variable. How strong a solution is a variable. So a controlled experiment is important because we're just changing one variable at a time. If we were to change multiple things at once, we don't know which variable is responsible for any change that we see. So back to that one we were just talking about with the plant stem, if we change the amount of light to see if the plants need the light for something, but we also changed the amount of water that we gave it, and we changed how much fertilizer was in the dirt, then we have no idea which one is making the plant stem move because we changed 3 different things at once. Just like if we were cooking a recipe and we added some salt, but we also took out the chicken and added some carrots and something about the flavor changed, we wouldn't know which one did it. So we only change one variable at a time. In a controlled experiment, only one variable is changed at a time. All other variables should be unchanged or controlled. So an experiment is based on the comparison between a control group, so something we don't change, with an experimental group. And I'll explain this in just a second. So we have 2 different groups, and they're exactly the same. We only change one thing. The control group is going to be the comparison. So it's going to be exactly the same as the experimental group, except one variable is being tested. So we'll have an example here coming up. So the experimental group will be able to show us how much that one variable is going to change the outcome, because that's the only thing that changed, and we still have that control group that stayed exactly the same. So our example here for what a control group is, because this can be a confusing idea for a lot of people. So in order to test a new vaccine, we would have 50 volunteers. That's our pool that we're going to get our data from. So 50 volunteers are selected, and we divide them into 2 groups, half and half. One group is the control group, and the other is the experimental group. So both groups are given this pill that has the vaccine in it. And the pill is identical in both groups. It's the same size, the same shape, the same color, the same texture. You couldn't tell the difference if you had one from one group and one from the other. So what do you think they're going to do with the control group? I want you to think about this in your head. How is the control group going to stay the same? So even though the pills look exactly the same, the control group is not going to get any medicine in it. It's not going to have any of the vaccine in the pill. So they're just taking a pill, and they don't realize that there's no vaccine in it. And we'll see what happens if they were just to take the pill without the medicine. So that's our control. That's the one that stays the same. Even if they think they're getting the pill with the vaccine, they're actually not. So what do you think the experimental group is going to be in this instance in your head? So this is the group that has the pill that has the vaccine in it. So it's the one that is experimented on. It's the experimental group. So they actually get the medicine, and we can see how much that is going to change the outcome of whether somebody gets sick or not. So what is the variable that we keep constant in this case? So the pill is exactly the same. The pill is just a placeholder in the control group so that they think that they are getting something, but we're actually just going to use them to see what would happen without the vaccine, because we don't know how much the vaccine is going to help if we don't have that baseline to base it off of. The control group is really just our baseline in any experiment to see how much the variable we changed is going to change the outcome. So what is the variable being changed here? It's going to be whether you got the vaccine or not, depending on what group you were in. So we have 2 different variables in an experiment. So we have variables we change. We have a control group so far. We have the experimental group, which is the opposite of that. But there are 2 names for the variables. So an independent variable is the variable that we change on purpose. So this is the one that we want to see change. The dependent variable is the one that you observe during the experiment. So the dependent variable is the data that we collect. And the best way to remember this is that the independent variable is the thing that we change. So in the case of the plant, it's going to be the amount of sunlight we would give the plant. That's the independent variable. That's going to change by us as the scientists. And then the dependent variable depends on what we're changing. The dependent variable is the data that we're getting. So it could be whether the plant turned towards the sun or not. Or if we're testing if a plant needs light, then we're going to test whether it dies without sunlight or not. So the dependent variable depends on what the independent variable is doing, okay? So in that example we had with the pill, what would the independent variable be? The thing that we changed. It's going to be the vaccine, right? We changed whether you had the vaccine or not. So one group didn't, and one group did have the vaccine. That's the independent variable. So then what was the dependent variable if the independent was the vaccine that we gave? What changed? What happened based on the independent variable? The health of the people that got the pills, because you'll be able to see whether the people who got the pills without medicine in it were any better, or whether the people with the vaccine were better than the other people as far as their health goes. So that depended on whether they had the vaccine or not. Okay, so once we get through the experiment, our next step is recording and analyzing the results from it. We have to collect the data in an organized way and then analyze it to determine whether it's reliable or not. So we're really figuring out, does the data support or not support the hypothesis? And it could be that maybe our experiment was terrible and we didn't get reliable information and we have to start over. Or maybe it didn't support the hypothesis and we have to figure out a new hypothesis. And that's where drawing conclusion comes in. So we look at that evidence and we use it to figure out if we proved our hypothesis or if we didn't. And it's okay if we don't prove our hypothesis, because that just means we need to look at our hypothesis again and figure out if we need to change it and design a new experiment based on that. We have to do experiments over and over and over all the time. Just because it might have worked the first time doesn't mean it's going to the next time. So scientists continuously are testing their results. They should always be the same over and over again before we can get a valid conclusion. Of course, we are probably not going to be able to do that in science class because we don't have the time to do that, but actual scientists will do that. So then we take that and we form a theory, and I'm going fast because I don't want to make this very long, but if you need to pause it, go ahead, go back and pause. Forming a theory. So after we do our experiment, we get our hypothesis proven or disproven, we can form a theory. So after it's been tested many, many, many times, and we have a lot of evidence to support it, it can become a theory. Now, in science, a theory is different than how we describe it in the real world. In the real world, we say theory as something that we don't know if it's true or not. It's just our idea. It's just our prediction. But in science, a theory is true. It's a big statement of what we have done dozens and dozens of hundreds, thousands of experiments on. And we think that this is what happened. So that's what a theory is. It's supported by tons of evidence. So a good example of this is the theory of evolution or the theory of plate tectonics. We say theory, and we have to remember in science, a theory is the truth. Okay? Okay, so we're going to go through the practice problem together. But this is the end of this video. Go back and make sure you have all of the notes if you need it, because this stuff is going to be super important as we go through all of our experiments. Thanks for watching.